Hi, I'm Srivarshini, a fourth-year bioengineering degree student from IIT Madras. In this video, I'll be talking about the use of graph network analysis to decipher the molecular arrangement of proteins. In diseases like Alzheimer's, Parkinson's, and type 2 diabetes, proteins tend to form aggregates that are very ordered. Our lab focuses on understanding the molecular pathway of aggregation in these diseases using computer simulations. A computer simulation gives us information about where these proteins are located at particular time points. Since aggregation happens over long time scales, we get data for thousands of time points. At a given time point, the system looks like this. And in the seemingly disordered system, if you look close enough, there are locally ordered segments. Finding these locally ordered segments and tracking how they change as a function of time gives us insights about the molecular pathway of aggregation. Previous studies have used a parameter called the nematic order parameter to understand the orientation of the system. This parameter calculates the average orientation of all the peptides in the system, and it works well only for systems with long range order. It fails to distinguish systems with partial order from systems that are completely disordered. Therefore, in a recent work, Charest et al. Have used an autoencoder to identify systems with partial order. But the problem is that autoencoders require huge training data sets, and its black box nature makes it difficult to obtain physical insights. Therefore, we have developed a novel graph-based method where all the proteins are assumed to be nodes, and proteins that are close to each other within 6 angstroms and that are oriented parallel are assumed to have an edge between them. And this translates the 3D data into an undirected 2D graph which have several connected components. Some of these connected components are indicated by boxes here, and each of this connected component actually indicates a locally ordered segment. Now, this effectively converts a complex, difficult to visualize 3D data into a very intuitive 2D graph. This graph also captures the actual connectivity of the proteins in the system. And now, using graph properties like degree, average shortest path length, we could classify these locally ordered segments into beta barrel, linear, or branched geometries. With the knowledge of these orientation of these segments, we can now go on to elucidate the molecular pathway and eventually find drugs that can target and cure Alzheimer's disease. Thank you.